I'm Amrutha, the SciTech Associate at GFI India. Extrusion has been around for a long time and has been used for food processing since the 1930s. For the smart protein sector, it is a unique texturization process where the feed containing isolated plant protein is subjected to high temperature and shear force that produces a final product containing an anisotropic texture similar to that of animal meat. Extruders contain a screw system housed within a barrel and a die, the latter of which is a key component used to provide the final product with the desired texture. Extrusion can be a low moisture or a high moisture process, and the most popular soy chunks that we've all heard of is produced by either of these processes. But high moisture extrusion using twin screw extruders is the most established technology for manufacturing third generation plant based meat products such as whole cut meat analogs. Therefore, extrusion technology is versatile and we can use different ingredients, both popular ones and unconventional ones, in varying combinations to produce uniquely textured products. At the same time, it is an intricate process and the parameters must be logically optimized in order to get the best product. So, to tell us more about this, we have an expert in the field today. But before we go into that, let me tell you about the GF Ideas India community. The GF Ideas India community is a community for innovators who are transforming our food system by creating or expanding access to smart protein solutions. The aim of this community is to advance the smart protein ecosystem in and for India. We host regular knowledge sharing webinars by domain experts, such as this one, and multiple social and professional networking initiatives. We also publish newsletters featuring deep dives across various topics in smart protein. We recently merged our SciTech India community and the GF Ideas India community to allow for greater diversity and have all our community members be part of a single large platform that provides more exciting opportunities. We'll be dropping the link for joining the GF Ideas India community in the chat box, so do keep an eye out for it. Now, who is this session intended for? Today's session is for students, scientists, innovators, entrepreneurs, and anyone passionate about smart protein who's looking to learn more about extrusion at an advanced level. For more information on extrusion, you can also take a look at previous GF Ideas webinars. We'll drop the link for those in the chat as well during the entire session. So for today's webinar, our organizing partner is the University of Transdisciplinary Health Sciences and Technology, or TDU Bangalore. And our speaker is Dr. Gurmeet Singh, Professor and Dean of Research and Outreach at TDU. Dr. Gurmeet Singh is a scientist, inventor, innovator, and entrepreneur with a rich career spanning over 3 decades in industrial and academic R&D. This includes his 20-year stint in the food industry at Unilever India and UK, during which he served as the Director of R&D. He's also spent over 10 years in academia, having served as an adjunct and visiting professor at several esteemed institutions, both in India and the US. He holds over 30 publications and 25 patents, primarily focused on tea processing and aroma recovery. Dr. Gurmeet is also the founder of Bev Science Private Limited, a knowledge-based tea and beverages company. In his current role as a professor and dean of research and outreach at TDU, he brings to life his passion for teaching and mentoring by supervising master's and PhD students and spearheading innovative academic and research programs, particularly in future foods, functional foods, personalized nutrition, and the texturization of plant proteins and meat analogs. Dr. Gurmeet's lab is home to the latest technologies and cutting-edge equipment relevant to food processing, particularly plant protein processing. We might actually get a glimpse of some of this equipment in today's session. Dr. Gurmeet is one of GFI India's key stakeholders and a renowned expert in the smart protein sector, having made significant contributions to the field of food processing and culinary science. We are honored to have him here today. Thank you for being here, sir. I'll now pass it on to Dr. Gurmeet to begin his presentation.
Thank you, Amrita, for that nice introduction. I need to put my slides on. I think you have to stop sharing and yeah. Okay. Where do I see my slides? Just give me a. Can you see the deck? I will. Is my deck visible?
Yes, Dr. Gurmeet.
Okay. So hi, thanks so much, Amrita. Thanks a lot to GFI as well for this opportunity. And welcome everyone. I see about 75 participants already there. So thanks for being here at 76 now at past 5 o'clock. So as Amrita mentioned, this webinar is focused broadly, right? Students, scientists, as well as entrepreneurs, but also a little advanced, right? Because we have presented the basics of extrusion a few times, right? And if anybody is interested in some of those, again, do write back. My email is there, and we can certainly organize other webinars as well, more basic ones as well. But we thought, let's step it up a little bit and see what we're doing, right, in today's world. Uh, uh, I have a few other people from my team. Abdul, many of you may already know, he's almost finishing his PhD, submitted his thesis, probably defending within a few weeks now. And as is an equal expert, I would say, has done more than 2,000 runs on the twin screw extruder, both HMMA and dry TBP. Manoj has also been around right from day one, has just finished his master's and is now beginning his PhD. Swasti has joined us from Guru Nanak Dev University, Amritsar, as a postdoc. She's done her PhD there, and Sahana is just embarking on a PhD and with interest in looking at mushroom proteins and combining them with plant proteins to look at what nutritional elements they bring in and what taste blocks like umami blocks they bring in. Uh, so yeah, so thanks to that team as well, because a lot of the data that I'm showing is from their team, largely from Abdul and Manoj, but the others are also beginning to contribute now. My institute, the University of Transdisciplinary Health Science and Technology, a very small university. We are about 40 faculty. We're about 10 years old, but we focus on a few areas, right? So we have largely focusing on master's program and PhD program, and looking at a transdisciplinary approach to health, right? And my particular interest is in an area we call as food futures, which is food first solutions to health. And health has three dimensions: human health, societal health, which is livelihood, and planetary health, which is environment. And the whole proteins area fits is a very important block, yeah, in there, because it ticks all the three boxes. Yeah. Rural India, RIST Rural India Supporting Trust is our big funder for the program, and a lot of the infrastructure, very expensive screw extruder, the rheometers, the RANSI mats, the texture analyzers, the headspace GC, the kitchen. They're all been funded by RISC, so we're very grateful to them. And GFI, of course, have been a partner almost from day one when we started our proteins program. And thanks to them that we have the visibility that we have today, right? Okay, so first question, right? I mean, a basic question, why proteins, right? We're talking about here texturization of plant proteins, right? And so why proteins? Why the interest in proteins, right? When we look at food, actually, proteins is not such a big component of food. 70 to 90% is water. Of the remaining 30%, about 50%, 55% is carbohydrates, 30% fats, about 10 to 15% proteins in foods, right? And then you have some fiber and you have the rest of the minerals as well. So proteins is typically the number four, right? Including water. And then many times it's the number five, right? In most common foods can bump up to in protein dense foods into number two as well, number one, number two. So why the focus on proteins is a question that all of us do ask, right? And there are many reasons, but I think in one line we can say that's because it has a disproportionate impact. No, so it weighs in a lot more than it weighs in grams, right? Both in terms of biological functions as well as the impact on the environment, right? Proteins are essentially, while people consider them as maybe structural elements, etc., I look at them as machines, right? Either the micro machines, which are the enzymes, or the macro machines, which are the muscles, right? And a machine's job is essentially that it has to do an action, a very specific action repeatedly, and that's what enzymes do, right? They perform like scissors, which is they will cut, or they perform like they pick up something and drop off something, right, at another point, like hemoglobin. So, and to do this, they require conformational changes, and that they do repeatedly. And more importantly, or more familiarly, the macro machines, not importantly, but is the macro machine part of it, which is the muscles, right? They have to contract, relax along the same length axis repeatedly over a lifetime, right? So they are machines in a way, right? So while they may be third, fourth, fifth from a food perspective, their biological function is huge, right? Because unless these movements happen at a micro scale and a macro scale, there is no living being. The second, of course, is the environment part, which is while a small part of our diet, their impact is probably the biggest, right, in terms of the environment, right? And therefore, there is this explosion of interest, right, that has happened in proteins and the way we acquire them in our diet, right? And here is a chart from World in Data. It's a very popular site. I love it. A lot of interesting graphics and charts there. And what it is saying is that, hey, about uh, half of the Earth's land, of the habitable land, is devoted to agriculture, right? 45-50%. And of that, a big chunk is actually 70-80% is for pastures and feedlots. So that's a lot of land that we devote to milk and dairy, which are two very dense forms of proteins, right, in our diet. And these contribute to about 40% of protein in our diet. While it seems a small number that 60% is coming from plants, but as you will see on the next slide, that uh, that not all proteins are equal, right? Uh, and they're not equal from multiple perspectives, right? Here is the one aspect of environmental perspective. There are many others, but we don't have time to go into that. That's for another deep dive lecture. Here I've just put some new data that's come out, which is around the land, yeah, part of how much of land, and that's the land area which is used to produce about 100 grams of protein, right, which is one serving. And we see in proportion terms that a lot of the plant-based proteins are in the bottom half, and a lot of the dairy and meat-based proteins are on the upper half, right? So overall, on an average, while we do see prawns at the bottom, we see fish, they're farmed. So those are the newer models which are coming up, right? But you can see soy protein pretty much at the bottom, tofu, uh, wheat protein towards the bottom, pea protein, groundnut protein, rice protein, grain protein, right? A lot of these proteins are towards the bottom half, right? So in terms of environmental footprint, land use, water use, greenhouse gases produced, the amount of energy required, right? Some of the data may be even more interesting than this one, but plant proteins score way better than some of the dairy meat proteins, right? That's the environmental. Uh, Also from a nutritional point of view, they are not equal, right? And here I'm comparing these proteins to a ideal protein, which typically is such as an egg protein, given by the FAO, the Food Agriculture Organization, or the WHO. And these are kind of ratios that you get. And you can see that a lot of, while almost everything in a dairy and meat protein will be 100% and above, right? Below 100 means that it's kind of missing something, right? That we see that most plant proteins stick on the lower end, right? They miss out on at least one amino acid, and these are the amino acids listed here are the essential amino acids, the amino acids that we cannot make ourselves, and therefore we do need to get them from our diet in adequate amounts, right? And we see that some of the sulfur amino acids, the SAAs, are marginally lower in the legume proteins such as soy, pea, chickpea, etc. And some of the cereal proteins certainly have a big miss on things like lysine, right? So that's why plant proteins nutritional footprint is lower, you would say, than dairy and meat, right, on their own, yeah, as a single protein, right? There'll be other data. The numbers will vary depending on what you've analyzed. This is one of our analysis, and this is the numbers we are getting. But batch to batch, there are differences and significant differences, right, that we see. So, so let's look at what are the challenges, right? While they're not equal, and the goal we keep hearing, and this whole talk today is around extrusion of plant proteins. So if plant proteins tick the box of environment but don't tick the box of nutritional profile, then why the interest in plant proteins, right? And secondly, are these the only challenges, or are there other challenges, right? And so I list a number of challenges which are there with plant proteins, right? Plant proteins are something which are very new to human diet, right? Humans have been around, Homo sapiens have been around for about 300,000 years, right? The primates have been around there for 10 million years, right? And we've had a certain diet. Uh, Plant proteins, dense forms like legumes, have only entered our diet over the last 8,000 years. So they're very recent in our diet, right? Whereas the other proteins, meat and dairy, meat certainly has been there ever since we started hunter gathering, and dairy proteins are again only about 10,000 years old in our diet on a regular basis, right? Uh, so plant proteins is still, while 7,000 years may seem like a long time, but it's still work in progress, right? For us humans to be able to eat them in dense forms on a daily basis throughout our life, right? And each one of those little adjectives that I have used, phrases that I have used in this sentence, are very important, yeah, phrases to look at. And I'm listing a few challenges here, right? I've already mentioned essential amino acid profile, not sufficient as a single ingredient, right? Uh, Digestibility, they are less penetrable matrices than, say, the meat proteins or the egg protein or the milk proteins, etc. Right? And therefore, they're not easy to break down completely in the time that they get in the small intestine. We still don't have great machinery to be able to do that, right? As a human being. The other one is less talked about, right? Carbohydrate load. This is that if you look at rice, no, are two big sources. People don't realize, but in our diet, if you also looked at the earlier chart I had mentioned, 40% of our proteins come uh, from animals, and 60% seem to come from the agricultural, the other foods that we grow. And a lot of that actually doesn't come from soy or legumes. Most of that comes from actually rice and wheat. They have huge amount of proteins in our diet. In Indian diet, probably 60-70% of our protein comes from rice and wheat. And so we get about 70-80 grams of protein per day. Many of us were consuming 2,000 calories. We do get that. But the challenge is that a huge amount of carbohydrate comes along with that. For every gram of protein, we get about 10 grams to 11 grams, maybe 12 grams of carbohydrates. Wheat about 7 grams. Legumes about 2 grams. So that's getting in the right territory, right? Because if you look at uh, uh, meat, I mean, 0 grams of carbohydrates, right, comes with it. And therefore, our ability to balance the carbohydrate to protein ratio in our diet is low when we have a lot of rice and wheat based diet. Yeah. The other thing which is even lesser spoken about is the phytate load, right? The 300 to 500 milligrams of phytate per 100 grams of legumes, right? And phytates are molecules which prevent absorption of nutrients like iron and zinc and other things, and some few of the vitamins, right? And maybe that is one reason why anemia is so prevalent in India, and mineral deficiencies are so prevalent in India. We've greater than 50% of the population. That's about 700 million people who are anemic at some point of our life, right? And that's because of this excess load. Similarly, the legumes are also rich in oligosaccharides like phosphorus and gauzes, fructose and glucose, right? And which not everybody can digest very easily. While we consider them as fibers and good for us, right? But not all of us can digest them in the amounts that they are there in legumes, in pulses, right? And therefore, daily consumption has always been a challenge, right? People give up eating a lot of legumes as you start getting to 40 and 50 and so on. Taste is always a challenge. Not all, certainly soy as such is not very appealing to this part, people who are unfamiliar with eating it. Some of the dals we are fairly okay eating with it. Uh, and texture is mushy, so you boil it and you get a mushy texture, and that's one texture, right? So very little play. Yes, we have a little bit of a dhokla that we can get. We can fry them and get some texture there, but quite limited as such, right? So what's the solution? Is there a solution? Well, the solution is that can we separate proteins from the other components which are there in the legumes and the cereals, right? And that way we can get a cleaner taste. We can reduce the phos or the indigestibility and make them daily proteins. We can lower the phytate levels to about 1/3, which is what the WHO recommends. We can really lower the CHO levels. We can uh, uh, increase, I mean, we can combine them to get quality essential amino acid profiles. So while we don't need to stick to one, we can combine them in innovative ways. We can process them for digestibility, like extrusion can improve digestibility, actually, make the matrix more available. Uh, we can texturize them, and so we can play with a lot of textures beyond the machinist. And we can probably make things which are affordable for masses, therefore, right? So, and those are things that we can start working on. And to do that, we have to get to what we call as concentrates and isolates, right? Therefore, right? And so if we begin with those as starting blocks, we are able to tackle and therefore create what we call as smart proteins, right? They are addressing so many challenges which are there with plant proteins in their native form, and we can solve so many challenges and make them smarter, right? Because this question should always be there, and you have to answer in your own mind, why do we call these proteins as smart proteins, right? What is it that's making them smarter, right? And it's being able to deliver on so many challenges together that make them smarter. So, okay, so how do we make these powders, right? Is the next challenge. How do we make a concentrate? How do we make a isolate? How do we take out some of the less desirable components so that we can get not just 60% of proteins, which is only worth half its value, but it becomes 60% of protein, plant protein in our diet, which is the value is 100% of it, right? So how do we make it way more meaningful, right, in our diet? And so there are two main principles we apply, right? One principle is the principle of solubility, to separate the protein part out from the rest of the part. And that's the principle that we have learned from milk, right? Where we make paneer, right? And paneer is basically we are separating the protein part of milk from the rest, right? And this is what we are trying to do here as well. We are trying to separate the protein part from the rest. But the first task here, while milk is already water, right? Protein and water. Uh, and we have to separate it from lactose, we separate it from fat, we have to separate from some of the other things, right? And 3% of all solids is protein in milk. We have about 5% lactose or sugar in their carbohydrates, another 5% fat. So we play with solubility, right? And solubility of protein is trying to either make protein more soluble or less soluble, right? Less soluble means it can be aggregated and make it settle down. While in milk, we only need to do one step because it's already solubilized or at least dispersed. And therefore, by changing the pH, we get the protein charges to neutralize so that they can come together and stick to each other and therefore aggregate. In the case of plant proteins, we first have to get them into a liquid from the solid state. So, and then we have to aggregate them. So this chart here shows the first phase, which is called the protein solubility chart and the pH. And we see that there is a certain point at which solubility is very high, which is say pH around 9 in the two extremes of the curve, around 9 or 1 or 2. And the solubility is low in the range 3 to 6, right, pH. So what do we do begin with? Because the protein is insoluble to begin with, it's in a solid matrix. We grind down the matrix and we change the pH to about 9 or so. Like milk pH to start off is somewhere around 7. In this case, for proteins, we need to get it to around 9 to solubilize it, right? So having solubilized it to 9, we have now been able to separate it from a lot of the other stuff, which is the carbohydrates, etc., right? But we've got other soluble stuff like minerals, etc., which have come out right into the liquid. So what do I do now? Now that I've collected a liquid which has proteins and has removed half the stuff which I don't want out of it, well, I now need to take it out, reduce its solubility, right? So I swing the proteins. First of all, I make the protein more soluble. When I have a liquid, my proteins are at about pH 6 to 7. I make the pH 9, swing it so that it becomes soluble. And once I have taken it away from centrifuged it and separated from all the other part, I bring the pH back to around 4, 5, so that the protein creates an aggregate, settles out separately, and now it's separated from the minerals, etc. Right? And I get a protein out. And this is what this kind of a flow diagram is showing. I took 100 grams of my legume powder. I put about 1,000 grams, 1 liter of water. I took some NaOH solution and I stirred it for some time, maybe an hour or so, and got the 25 grams of protein which are there in this 100-gram legume out into the water phase. And then I filter it out or centrifuge it. The supernatant, because the protein is soluble, is the supernatant. The liquid part has most of the protein, I hope, very little protein in the sediment. I collect the supernatant. I now swing the pH back to about 4, so I'm adding HCl. In this case, you could add any other acid that you wish, any other food grade acid. Our stomach makes HCl, so that's a real food grade acid that we put in. It's basically stomach acid, as we call. And then we centrifuge it again, and this time the protein hopefully is aggregated because it's lost its solubility at the lower pH, and it will sediment out here, right? So that's the overall process flow that we get into, two simple steps, right? And here we see what's happened. I had 23 grams of protein about in my 100-gram mung bean powder. When I collected my first supernatant out of this, I got about 19 grams protein in my liquid. My 3-4 grams I lost in the solid part. I couldn't get them out in the single extraction that I had done. And when I precipitated this out, I got about 16 grams recovered here and 2 grams I lost in the liquid part. I couldn't settle them out, right? So out of the 23 grams, I got about 16 grams. So I got about a good 70% recovery. Not bad for a simple extraction. If I had done double, I could have gone to probably 80, 90% extraction efficiency. And the other is looking at purity as such. Total weight of my pellet was about 20 grams. I had started with about 91 gram powder. I got 19 grams. So 16 of those 19 grams were protein, which means my purity was of the order of 80, 85%. So good. I had 70% recovery and about 85% purity. So that's a simple experiment that we do, and we have the whole lab set up to be able to do this. The centrifuges, the spray dryers, the extraction vessels, the homogenizers for the ultrasonicators, if you want to use that to be able to do this. So at a bench scale, we are able to get good quality protein powders and then look at the quality of the protein, their amino acid profile, the length of the different kinds of protein through an SDS page, the physicochemical properties, and that then allows us to fine-tune conditions and then maybe go to a larger scale somewhere and extract proteins and test their texturizing ability or their ability to go into plant-based milks, right? So that's one, the liquid extraction. The second is way to extract protein powders and separate them from the other lesser desirable materials is to go to a process which has emerged recently, become popular recently. The process been old is the dry concentration method, right? So what is dry concentration? This relies on what is the principle here? The last principle was solubility. Here, the principle is a physical principle of hardness, right? In a dal, in a legume, when you crush it, we said there is protein in there and there is starch in there besides other things. So there's basically starch, right? 50% of legume is starch. And then there is the non-starch stuff, which is the mineral components, the protein components, some fiber components, right? So the starch granules are actually very hard balls, like golf balls, right? If you look at a micro scale. Uh, the protein part is softer, right? So when you grind something down, uh, you get you don't get a particles one particle size. You get the whole size distribution. And what we see is that the coarser sizes, if you sieve them, they are richer in starch and poorer in protein, and the finer sizes are poorer in starch and richer in protein. And that's because starch granules don't break down easily. Now, of course, if you use a lot of power to break down, you will break down starch granules also, and then you'll not be able to separate through this difference in hardness, right? So you have to optimize what kind of force and energy you are using, what kind of attrition you are using to grind the particles, because you want to make sure that you're not breaking down the starch granules, right? That's the key to this. And therefore, there is some little work which is required. And you play around with operating conditions to make sure that you're not breaking starch granules. And if you do that, you get a nice separation. And you can go up from about 20-25% protein in a dal mix powder to about 50% or so protein in the finer part, right? Your recovery is also about 50% or so. It's not great, but you've got 50% recovered for at 50% concentration. And it's a straightforward process, right? Straightforward because you can look dry fractionation, milling operation, and then classification, which is actually the separation. The physical sieves don't work there. You have to go to an air separator, air classifier to be able to separate the sub 20 micron particles from the above 20 micron particles, right? And you collect two fractions, the fine and the coarse fractions. Whereas the wet fractionation, as I said, you have an alkaline extraction, and then you have an isoelectric precipitation, you have multiple centrifugation steps, you have spray drying till you get the isolate, right? So this is a number of many more unit operations, where this is just two unit operations. You grind and you separate, right? But of course, there's a difference. Here you can go to dry fractions, you go up to about 45, 50, 55%, and you have 50% recovery. Whereas in the other one, you can go up to 95% if you are working with soy, and you can get up to 90% recovery there, right? So there are different. And so what people are beginning to do is go into what we call as green separation. That the first stage we do a dry fractionation, and then once we have got the concentration up to 50, we do a wet fractionation. That uses way less water, way less energy over the whole thing, and it becomes a greener process, right? So that's the fractionation part, right? So now, of course, we've collected the powders, and we have more desirable powders, in the sense that the phytates, the carbohydrate load, some of the other factors, the undigestibles, non-digestibles are out of it. And we can now start mixing these, right, in the right proportions. I can take a soy protein and a rice protein put together and go to a very different kind of a functionality. I could also put in algal proteins, I could put mushroom proteins, I can put the fermentation microbial proteins in there, and really improve, bump up the profile, and even try and aim it to get better than milk and meat proteins, right, and egg proteins. And that's one area where I don't see a lot of startups working, right? That where can I take some of these fermentation proteins, combine them with plant proteins, and can I bump up the quality way beyond what meat proteins can do on their own? So I think that's the area that you have to start working towards. Uh, then, of course, the next challenge is that we want to create a variety of textures. And why do we want textures, right? I had a slide which I've taken out, I think lost somewhere, but that's because, once we humans get teeth, we start getting at the age of 1, by 2, 3, 4, we have a good set. And by adults, uh, we have a strong set of teeth, right? By the age 12, 13, and so on. Uh, we eat food. Yeah, we do drink some food, but largely we eat food, and eating is all about textures, right? So we are tuned evolutionary to love textures, right? And while you will hear taste spoken about a lot in food industry, I spent 20 years in Unilever, and a lot of our focus was taste. Texture is actually even bigger than taste, right? In our mouth. So, and therefore, we are trying to see now that we've created good quality protein powders in all respects, nutritional respects and other respects, how do we texturize them? Because plant proteins, as you said, are mushy. If you heat them, you put water, they will become pasty, right? They don't become fibrous. So we want to get to fibrous textures because, well, pasty is fine, sauces and other things, but fibrous would really open up the range of products I can create. And so that's a challenge that I've been working on, right? And uh, and to create these textures, unique textures, so therefore what we want to do is we look at what a protein, plant protein looks like, and we see that, hey, like most proteins, plant proteins are also interacting. That whole chain is kind of wound up together into a ball, and that ball is not easy to open. It's stuck, and it's stuck to create that shape because of bonds. And we call these bonds, as now we understand them well, they are hydrogen bonding, they're hydrophobic interactions, they're disulfide bridges. And if we really want to create textures, especially fibrous textures, we want these balls to open up and start interacting with each other's strands, to interact with each other in different ways. And therefore, we need to break these bonds, right? And then be able to rearrange them in a different way. And how do we do that? We have to use heat and we use shear, right, to do that. So heating is clear that if I heat, I can break these bonds. That's very clear. And this other picture talks about shear, that if I actually put force on this, a force field on this, which is parallel to the surface of the protein, I can start orienting. So this is the microstructure at rest, and this is the microstructure under shear, right? And you can see, if you look at the protein, which is this figure, the rightmost figure on top, right? I can, once it is opened up by breaking by heat, I can start aligning these together by introducing shear, right? So what I'm looking at is that if I have a controlled program, right, in the language of food processors or food technologists, it can create a controlled program of heat and shear, right? Thermal field and force fields, right? Then I can arrange these proteins in novel ways. Yeah. And after arranging them in novel ways, if I can get them to bond, to stick together in the new configuration that I have created, then I will be able to create stable textures, which will stay like that, right? Through cooking experiences. And so that's what we want to do. We want to introduce a very controlled program of thermal field and shear field. And how do I introduce this control, right? And for that, the one equipment which allows us to do that in a continuous manner and at scale is called a twin screw extruder, right? Because it has, number one, that along the length of this, I can program a temperature profile going from one end, let us say this is the feed end, to the discharge end. I can create a temperature 30 degrees, 50 degrees, 70 degrees, 90, right? And so on. I can create a thermal program, a heating program, right? A heat profile. And similarly, this is a cross section of this. And what it is showing is this is the wall, the outer circles, right? Two screws, and therefore it looks like an 8. And inside are the two screws which are perpendicular to each other. This whole white thing is a screw. And the whole colored portion, the blue, the yellow, the red, right, are the shear fields, right? Where the shear is the highest, wherever the gap between the screw and the barrel is the lowest, and that's the yellow part. So it's highest here and some high fields here. Blue is where the shear is the lowest, and red where it is intermediate. And I can arrange these gaps between these by selecting different shapes of my screw elements, right? And I can also organize the screw elements very differently along the length. Here it seems to be the same screw element, but I can keep changing them along. And therefore, I can change and program, as I call, the the shear profile. And that's a big part of our program at TDU, is to play around with these thermal fields and shear fields and understand what that will do to the nature, of this product coming out, to understand those force fields. And right. So, by combination of these two, the mixing elements and the heating elements, and here you can see a picture, right, of all the different kind of screw elements, as we call, right, which are there, which I can load onto a shaft like this, and it's almost like putting bangles onto my arm, right, where your arm is the shaft of the screw, and then these screw elements, and each of them will introduce a different shear field and a mixing field, and therefore I arrange them, about 50 of them, along a 1 meter of shaft, and create my force fields that I want. And similarly, I program the temperature, and that gives infinite combinations of shear and heat in a twin screw extruder. And that's the flexibility, right, which has made this instrument very popular, right? And this multiphasic protein system can then be structurally rearranged to impart what we're looking for is an anisotropic. Anisotropic is that when you cut something along two different directions, its physical properties are different. So cutting strength along one direction versus the direction perpendicular to it, if they are different, it's anisotropic. That's what wood is, right? If you cut along the fiber, it cuts easily. If you cut across, it's more difficult, right? So we are trying to create these fibers. So, and we create different kinds of structures, right? And depending on how we run our extruder now, right? I could do a low moisture extrusion. So the earliest extrusions were expanded, right? We're using expansion as a texturization method. And to get materials to expand, you want water to evaporate. You want a big pressure drop, right? So if you require two things. One, you require a something which will cause the expansion. Water is a very good thing because water at high pressure will be a liquid, and once you reduce the pressure to atmospheric and the temperature is above 100, it will turn into vapor, and that's a volume expansion of about 1000, right? You go from a density of about 1 gram per liter to about 1 gram per milliliter to about 1 gram per liter, right? So 1000-fold expansion. So that's what this tried to do. We created high pressure conditions, about 100 bars, right, in there, and a moisture of only about 20% or so, and a dye of about temperature about 150. Why I want a low moisture? Because if I put too much of moisture, it will cause a lot of slippage and my pressure will drop down, right? So to get to a high pressure, I have to operate at a lower moisture. I do want some moisture because that's the water which is going to evaporate, right? But I can't have too much of it because then I will not get the pressure that I want. And I want the temperature to be high enough because that also helps me in the phase transition and also helps build the pressure. So those are typical conditions. And if I do that properly, I can get a nice expanded structure. My key there is not expansion because I don't want a nugget kind of a spongy texture. I want a little dense expanded matrix, which is why I want a twin screw extruder, which will provide me density as opposed to a single screw, will just provide me plain and simple, very low density expansion. And and I want good hydration characteristics, however, right? Because I want it to be dense, expanded, but dense. But I want it to be able to hydrate as quickly as possible because in industry, I don't want to be sitting around for an hour or 2 hours for it to hydrate. Also, I want to hydrate in room temperature water or cold water if I'm going to be using it for sausages, because I use the next operation is iced operation, right? That I use in my bowl chopper. So those things start playing, and that's what a lot of the experimentation is around. How do I get a dense expanded matrix which will hydrate, however, very quickly, even in iced water, right? Depending on the application. Whereas high moisture is about getting even further high density. I don't want expansion now. I really want density like real meat, right? And therefore I want zero expansion. So while low moisture expanded products are good for structured meat or minced meat kind of applications, or the high moisture ones are more suited to shredded meats and things. So this is like the holy grail we are all after. I don't think it has been perfected anywhere in the world yet, because we do uh, tend to be a little more chewy than we would wish to be, a little more cohesive strength, then I would like the meat to fall apart more easily than it does here. I can do some more use with low moisture extrusion and shape them to look like these kind of meats, but on their own, we are not able to do that globally. We are getting there, but only halfway, I guess, right? So to be able to create these dense structures, I don't want too much of pressure drop, right? When the material comes out of my die. And so less than 10 bar pressure. I don't want the temperature to be too high. So about 110, 130 is where I operate. And how do I keep the pressure low? I put in a lot of moisture because moisture will cause a lot of slippage, right? Make sure my pressure doesn't increase, right? So 60, 50 to 70% moisture typically. And here is the design. I have a feed element here. I have my water introduction, my screw configuration, my heat configuration that I've provided. And what I want for my high moisture is a long die because I want to bring the temperature back to about 60, 70 degrees so that there's zero expansion almost coming out. And also that temperature will allow my material that I have created, the rearrangement I have created of the various proteins after opening them up, they get to stick together into a fibrous structure, right? So that's what I'm looking at, and therefore my die is here. It's almost doesn't seem that big, but in many cases this die is almost as long as the screw, right? That you would like. So, okay, overall, what have we got? Therefore, in a twin screw extruder, I said I can decide on my protein source. I typically use a protein blend. I don't use one because, as we said, we want to get the right nutritional profile. So I'll mix a few proteins to get the right nutritional profile, but I also add some other components to make sure I get right textures, and I'll come to that a little bit later again, right? Then I need my equipment configuration. What is my screw type? What's my screw configuration? Because I need to program my shear profile, right? And then my process parameters, I profile the temperature profile of the barrel, my die temperature profile, and I fix my feed rate and RPM, right? Those are things that I set. And at the other end, I'm looking at what is the hardness of the material, what is the cohesiveness, what's the springiness, the resilience, the chewiness, the cutting strength. Those are my consumer facing attributes that I'm looking at, right, for my materials coming out. And as a process technologist who's working on the line, what are you looking at? These are the intermediates you are interested in, right? The left part, a technician will set, right, on the machine. The right part, you have the parameters that you'll send to a test lab. But as an engineer, you're working on these, that how do these parameters end up in what's the torque, what's the pressure profile, what's the residence time, what's the kind of mixing, what's the thermal exposure, and we are interested in these things, measuring these things as a scientist, as a technologist, as an R&D person, working on trying to create the perfect texture for your product, right? To be able to understand what's the expansion I want, what's the vibration I want, what's the air entrapment I want, what's the viscoelasticity that I want, no, that will cause the consumers to think of it as whatever product I'm trying to mimic, what the gelation, what's the emulsification that will it hold oil, will it not, etc. So those are some other parameters which start getting you closer to this space, right? So that's the journey that you're on, and therefore we are trying to measure everything around here, and that's what we have in our lab, that for each of these things we have an instrument to measure, right, these things, uh, and that allows us to therefore understand what we are doing, right, to the level that we wish to, and be able to then scale it up and repeat, and then be able to create a lot of different textures and colors, etc. You can see the density of fibers varies across, the length of fibers varies across. These are very fine fibers oriented. These are very coarser fibers that are there, right? So I can play around with the density of fibers. I can play around with the thickness of fibers and the shape of fibers. So I can play with the visual appeal. But as I said, I've not taken powders. I have also now started structuring them. Now, what I want to do is to be able to understand what happened right in there, right? And there are two types of tests that we do, right? Uh, Two physical tests, and then we do some tests to try and understand what kind of forces were used in trying to put this material together to structure the material, what kind of bonds, what kind of chemistry happened, right? So to understand the physics of the problem, we look at what are called as compression tests, which is that we press, right? The material, I take the material and I put some force on it, and I measure how much of force I put and how much the material deformed, right? And that is a compression test and tells me about the structure, right? Similarly, I could also do a twisting action, right? Which is that I put my material, rather than pressing it, I let it touch and then I twist it, right? And then depending on how quickly I twist it or how much I twist it, I start getting some very interesting information, right? And why are these two tests important? Because, if you put a piece of food in your mouth and you are biting into it using your molars, let us say, or premolars, right? You're doing two actions. One, you are pressing it, and that's a compression test. But you are also kind of twisting it, right? In the sense that your molars are not moving just down, but you're shearing it, right? From front to back also, right? And that kind of action of your molars, this is the non-compressive action, but the shear action is captured here. So this is more of the shearing action of the molars, and this is more the compressive action of the molars, right? And then using a knife kind of an assembly, I can also do the biting action of the incisors, right? That can happen here. So you can imagine that I'm trying to understand what will happen in the mouth, right? Through various experiments, through physical measures, right? And there's a lot of math and science which has been perfected over the last, I think, nearly 40 years now, to be able to relate this to consumer terms, right? Also. So I'll skip this part, but this is the consumer part of it. This is just a force, right? Versus distance curve, right? That I'm pressing a food and what happens, right? And different parts of this curve. So you can see that as I press, I require more and more force, and then I get a peak force at which probably the material kind of breaks down. And then as I move my jaw back up, the force starts reducing because I'm pulling my jaw back up. You could think of this as jaw being moved up, right? And then I come down for a second bite. And because my material has probably moved down a little bit, maybe this distance 2 may be separate from distance 1. This may be a smaller distance. It could be the same if it's bounced back entirely. And maybe I require a lesser force to now get it down, right? Break it down and then, bring it back up. And the various features of these physics curve, force versus time, for those of you who remember your high school chemistry, the area under the curve of force versus distance curve is the work done, right? That's the work done by my jaw, right? The peak of this curve is kind of the hardness. What is the maximum force before a material breaks down, right? That's related to hardness. There are cohesiveness, how cohesive the material is. That's the area of the second curve versus the first curve. If the area required, which is the work done, is the same in both, which means my material is absolutely cohesive, right? It's like a gum or a rubber which I press down and comes back up, and I press down and comes back up entirely, right? So that would be a very rubbery material, right? Springiness is whether the material stayed down, right? If distance 1 and distance 2 are same, which means it bounce back to the same level, right? It may be broken inside, which is why my cohesiveness is not there, but it just jumped back. So it tells me some springiness. Resilience is how much of force did it require for the material to go down and how much did it push back up, right? The material is also pushing my jaw back up, right? And so by comparing the second half of the curve versus the first half tells me about the resilience, right? And if the curves are uniform, this one is not, right? Which means it has low resilience. If they're uniform, which means they're very resilient, right? And then chewiness, etc. You can derive various other parameters, gumminess, stickiness, etc. as well. But it's a very interesting use of physics to understand sensorials. And that also tells about how much of viscoelasticity, the rotational part tells me how my material is, how much of it is, what is the viscoelastic characteristic. No, the visco characteristic is much does it behave like a liquid, and the elastic is how much does it behave like a solid, like a spring. And all materials are somewhere between viscoelastic, right? And also tells that till what strain, how much of deformation will it take, and where is the linear region, and where does it start breaking down, and maybe the solid starts shifting toward a liquid kind of a texture or broken down. So those are various measurements that we do. And this here shows that if I started off with a soy, which is what the topmost line is, how long is the linear material. And if I start adding another material here, I've added millet flours, and I can see that the curve starts going in one direction. The breaking down as I blend more and more of it, right? The transition from a solid to a liquid, from a elastic material to viscous material happens a little earlier, right? At lower strains. Whereas if I added wheat to it, the transition happens at a higher strain to it. So I have to press a lot more or chew a lot more, provide that shear action in my mouth a lot more if I start adding wheat blends to it. So we can understand by adding another component, how does the material start changing in the mouth, right? So uh, in our lab, we do something which is called a full spectrum analysis, which is we don't just look at hardness. So this is a comparison of how the soy protein high moisture meat analog, how much its hardness was, chewiness was, resilience, cohesiveness, etc. And here are three meats, right? We had a chicken, we had a lamb, which is a green curve, and we had a beef. And we say that just 100% soy or high moisture were way beyond. They were way harder than real meat. They were way more chewy than real meat, right? And really didn't feel like on their own like real meat, right? While they were still edible, I could use them, but they were out of spec, right? So we would like to get them, play around and see how do I get them right into the meat territory. So we play around what we have. We play around with protein levels, moisture levels, the feed rates, screw rates, barrel temperatures. As I said, I can play around with the shear profile, temperature profile, right? A lot of different experimentation that we do. And we can see that I can create hardness levels going all the way from here to here. And we do understand really well as to what's the impact of each of these parameters on hardness, right? What does a big impact, of course, is the protein level in there, right? We've gone from 18% to 28% protein on a wet weight basis. We feel the wet weight basis is a lot more meaningful than just a dry weight basis, which is where most of the literature is. So we've been pushing for wet weight basis in literature, not so very successfully yet. But hopefully in the next few papers, we may get that point through. But we do see, as you know, it's 18%, 21%, 24%, 28%. You can see it going up. And then within each, you can see the effect of what does the temperature do, what does an RPM do, what does the moisture do, right? What does the feed rate do, and how you can play around that little space once you have your moisture and protein content done, right? How do you play around? And given the space, the parameters we play with, each dot is a different run, and we can see that hardness, chewiness we can get within the range, right, for these the chicken, lamb meat, and beginning to close the gap with also resilience, cohesiveness, and springiness, right? We're not there, but as I said, that's work in progress, right? And we published this in one of our papers, which came in the Innovative Food Science Journal, right, in Elsevier Journal last year. Uh, has had almost 20 citations now, I think, already. Uh, so that helps us in physical understanding. I also mentioned a chemistry understanding, right? That what kind of bonds came into creating those aggregates, right? Now that I understand how it's behaving, how much is the solid, how much is the liquid, what forces are required to deform it, what came together to create this structure from a powder, right? And that we wish to understand. And for that, we use the understanding which has been created by the protein chemists, right, where they have created a number of reagents, right, to break various kinds of bonds, right? So a covalent bond like a disulfide bond can be broken down by mercaptoethanol. Uh, a non-covalent hydrogen bond can be broken down by urea. A hydrophobic bond can be broken down by thiourea or triton, or right, and so on, right? So we said, okay, let's start playing around with these reagents, and let's see how much of protein solubilizes, right? So we created a set. We take a protein sample, something which has come out of our after texturization from our extruder. We grind it down and we add our various solvents, right? Protein solvents, from the standard phosphate buffer to SDS page or a urea or a mercaptoethanol or combinations of these, right? And we are beginning to become a Sherlock Holmes, right? That, okay, if I use this combination, I should be able to break hydrogen and hydrophobic bonds, but not disulfide bonds. If I use the PSM, the phosphate buffer, the SDS page, right, etc., I'll be able to break disulfide and hydrophobic but not hydrogen bond, and so on. And therefore, by using various combinations of these and looking at protein extractability, solubility, we become the Sherlock Holmes of the protein world and try and understand what bond. And this chart here is trying to show that we have four materials. We have the raw protein powders, and we have the high moisture meat analogs with soy, and this is with a millet blend in there. And we can see the solubility with just a phosphate buffer is very low. If you use all the reagents together, the solubility is high. And by looking at what are the differences in solubility with each buffer, we can begin to understand what kind of bonds have come into play, whether it is the disulfide or the hydrogen or the hydrophobic, right? So that's a part. There are other, so we've spoken about protein blend principles from a nutritional perspective, where I showed the essential amino acid profile. We also look at preparing blends from a physicochemical perspective, because if you're looking at fibers, visual fibers to be created and anisotropic structures, what we realize is that we require two components. If the two blend components that we take are way different in their physical attributes, physicochemical attributes, especially their water soluble, water absorption, then we get very nice thin fibers, right? And therefore, you can see here, SPI has a very high water absorption, almost 6 here. Wheat gluten has about 2. And if we combine the two together, it's a difference of nearly 4. I get very good fibers, right? But I'll also get good fibers because rice protein is also a difference about 3.5. Mung bean also about a 3.5. So I start getting good fibers there. I've not put starches here, but a lot of the starches are also in this space about 2, etc. So by adding those, I can get to good fibers as well. But of course, if I add starches to that level, I will start losing out on the total protein content of it, right? So we have to balance all these things and come up with the blend. Therefore, how much of primary protein component, how much of secondary component? Is that secondary component only another protein, or will it be a starch? And therefore, what should starch be the tertiary component? How much to add? Which protein to add? The nutritional algorithm will kick in. So you have to put in multiple. So therefore, it becomes an algorithm as to where do you want to end up with, right? Then if you want to get to oil absorption and juiciness, because you're going to finally use this in a recipe after marination, you have to start getting into oil absorption versus water absorption also, and start putting that into your blending algorithm. And then emulsion activity and foaming capacity. If you're certainly more from, if you're making plant-based milks, because we also have a whole plant-based milk laboratory that's lesser known. Most people think of us as extrusion laboratory, but we have very good homogenizers, high pressure, high shear, and the capability to make very good plant-based milks as well, right? And there also we play around with what are the characteristics and make a selection, right? We played around with almost 20 different proteins in the laboratory. I'm showing 5 here. RPI means rice protein, WG is wheat gluten, CPP is chickpea protein, PPI is pea protein, and SPI is soy protein, right? I should have given the full names. Yeah. Then we also have viscosity characterization that we do. So this is what is called as a pasting curve. Of course, there is no pasting in protein isolates. There, most pasting curves are usually associated with starches. But you can see under similar regimes how viscosity starts building up, and how soy behaves so differently from anything else in the viscosity, which means it starts creating a network, which is why viscosity builds up even at a low temperature. The dotted line is the temperature profile, right? So this is a total experiment of about 21 minutes, where we did a temperature ramp up, which is the red dotted line, almost up to 90 degrees centigrade over 9 minutes, held the temperature there, right, till about 15 minutes or about 6 minutes, and then brought the temperature down slowly over the next 6 minutes or so, right, back to about 50 degrees centigrade, right? And we kept rotating measuring viscosity through this time, and the viscosity at each of these temperatures is therefore recorded, right? And we can see as the temperature goes up, the material starts melting, right? Going from a solid to a melt, and so the viscosity should go up, right? So the viscosity does go up. And then we are holding it together, and we see the material starts relaxing. And then as it cools, the viscosity starts building up again, right? But interestingly, wheat and rice, they do not have a change in viscosity. They don't melt very well at these temperatures. They require higher melting temperatures also. And therefore, they're more solid at that temperature, will not create good networks there, right, in these conditions. So we get some sense of all the material. So finally, my last two, three charts, time to end. What are the learnings, right, that we have? Firstly, of course, simple learnings. Not all proteins texturize is what we have, right? Soy is the best. Pea, chickpea, mung are okay, right? Uh, so soy is also the lowest cost. So you have to have a really good reason why you want to use something else. Uh, Rice, cashew, etc. We're not able to texturize, but they do bring in very interesting nutritional elements, right? We can incorporate them at 30-40% levels and still have very good amino acid scores and really interesting textures, right? Uh, Concentration effect, high protein concentration, very high, make very hard textures, right? So nobody would ever use an isolate 90%, right? You do need to get to about 70% or so. But if you use a concentrate, you have no control over what else that material has. It may have a lot of anti-nutritional factors. While it will texturize very well, right? It may not be your daily protein yet, right? So there is a, should I spend that extra money for isolate versus a concentrate? Is a decision you have to make, that if you want somebody to have on a daily basis, I strongly feel isolates probably will do the job better, not because just texturization, but because of some of the other components which are still in there. Fibration, what we have realized is I can get good visual fibers, but that doesn't necessarily mean I'm getting uh, the cutting strength anisotropy, right? And therefore, you have to work on both, right? You will get a good images, uh, and you will also get good XRD images, etc., showing fibration, but you will not get good cutting strength differences. And so, in the mouth, no matter what direction you are chewing, right, they don't behave differently, which seems a little odd, right? So that's something we work on in our lab. Full spectrum texture matches are difficult. As I said, hardness, chewiness, easier. We've been able to get resilience, springiness, cohesiveness, juiciness, a little more harder. We're getting there in high moisture meat analogs. When it comes to low TVPs, no, the dry TVPs and then structured products, no challenge, right? We can excel there in our lab. The high moisture, I think nobody in the world has kind of cracked it fully, right? Blending rules, of course, amino acid is the algorithm one, but we now have physicochemical algorithms also, right, that we are beginning to understand to come up with the blending rules. And there is, of course, always a need to understand more the system to process. And that's what a lot of our team looks at, that given a powder, can I actually just run a few tests, rheology tests and physicochemical tests, and be able to understand what will it do in the machine, because machine runs are expensive, right? So how do I predict that? And therefore, when I am doing my extraction and preparing my concentrates, isolates, and powders, can I start taking those results upstream and modulate the protein profile there itself, right? And relate texture to the other parameters. Uh, we do have some publications, two publications in good journals, I think very high impact journals. Uh, and one report that we wrote with GFI, GFI wrote and was a consultant there. And we have, I think, one publication ready for, and one patent that is getting drafted. And I think other three publications are in the writing phase, right? So hopefully this year you should see a lot. We also have one review article which just got accepted, which with GFI, Padma, and with Brabender, Devomitra, which is a Brabender scientist in the US, and our team, we've co-written the extrusion of millets, no, in meats. And so that should be coming out very shortly. So yeah, so that's where we are. I'll stop here. And saying, if anybody, because I'm hoping there's a lot of student population there, if anybody is interested in an MSc by research, do contact me. If you're interested in passing on the word to BSc, we have a BSc food processing technology program as well. Please pass on the message to them. And people interested in doing PhDs, please feel free to contact us. PhDs are always limited space, but we'll always look at how you can join us there. So yeah, with that, I stop. And any questions? Yeah.
Thank you, Dr. Gurmeet. That was such an insightful session, and I definitely learned a lot. It was really interesting to zoom into all the details of why protein and how do we do that. So, very, very interesting session. Now, let's just move on to the questions. We have about 10 minutes. We can explore some questions now.
Yeah.
So, Ayush asks how to control loss of nutritional value over denaturation of proteins during the extrusion process.
Well, denaturation does not reduce the nutritional value of a protein, right? The all proteins are broken down in our digestive system before they're absorbed, right? So our stomach, and intestine, they are going to be denaturing the protein, not just denaturing, they are going to be hydrolyzing it and bringing it into free amino acids, right? And by denaturing in a extruder, there are reports out there which actually shows that it helps the stomach, right? It improves the digestibility. So let's not look at denaturation as a reduction in nutritional value. The nutritional value of a protein has two important elements in there. First is that what is the amino acid profile, and all of it is an L profile and not a D profile, right? Because L to D can happen, but that doesn't happen in the conditions we use. So that's one. And second, is the protein available for our enzymes to interact and break it down? And yes, it is, right? So so that sense, yeah, we shouldn't worry about that. Yeah.
Thank you, sir. Another question from Pranab here. Sir, have you done any study on dry fractionated concentrate? Can you throw some light on applications in TVP and HMMA?
Yeah, so dry concentration, we have a equipment which is under order. Hopefully it'll get installed. The dry concentration unit will get installed in July next month. We'd ordered it in January. It should have been installed in May, but there's a little bit of a delay from the manufacturer end. But there's a lot of literature, right, in that space, right? And we've done some basic experiments at very small scales, yeah, to look at that. We've done experiments at other third parties, right, where we got good results, right? Especially where we ordered the equipment, we did a few trials there, right? So, so yes, positive results. As I said, the limitation is on that can take you up to about 50% protein, right? 50, 55, 45 to 55% typically, from 20, 25%. So yes, doubles the concentration. And therefore you can start doing a number of things. You can certainly start texturizing a little bit towards in the expanded space. You can't do high moisture meat analog, for which you require protein concentrations in the range of about 70%. So you will have to do something else, but it can become an important blend component along with the isolate, right? To put in, brings in a nutritional profile, right? So there are certain advantages that it has. And so it's an important ingredient in the set of ingredients we have, right? We are always looking at what's the toolbox that I have, right? And having, or the ingredient box, and having a dry concentrate is a very important part. Yeah.
Okay, thank you, sir. Another question from Kishore here. What are all the difficulties in using millet-based proteins or blends as primary ingredients in an extrusion process?
So we've not used millets as millet proteins, right? Millet has only about 10% protein, right? Again, not great quality. I mean, I have, and nobody has really, there are not enough processes around millet like rice and wheat. No, wheat starch is a very big industry, and therefore wheat protein is available. Wheat gluten is available at a very low price. Rice is in use for ethanol production, right? Bioethanol production. And therefore rice protein is available at a low cost, and both of these are available in ample quantities. If you make a millet protein, what do you do with the remaining 90% is still a question. So I think before we get to millet proteins, there needs to be research on what do you do with the remaining 90%. The reason soy proteins and wheat gluten and rice proteins have picked up so much in pea proteins, because people have figured out what to do with the remaining stuff. In the case of soy, it was oil, right? In the case of wheat and rice, One is no that you are using the starch part or using the ethanol part, or right. So that's how they have become useful. If you're using millets, right now there's no use case for the rest, so it will be of academic interest, right? We haven't even separated the proteins yet. Millet proteins will be more challenging than the rest because there are a lot of polyphenols and other stuff, right, which are there in millets, which are easier to handle in the other cereals compared to millets. But having said that, millet flours have their value, right? As I said, you have to introduce a second ingredient into an ingredient which has very different physicochemical properties compared to your primary protein ingredient, to be able to manipulate the fiber profile, right? What can you improve the number of fibers per unit per cross-sectional length, right? For example, right? And those kind of things, different ingredients bring in their own characteristics. They also bring in their nutritional spikes in different directions, besides the textural element. And that's why we use millets, right, in there. It adds to a ingredient. We feel that when we use pure starches, we are adding a almost like an E number, a very highly processed ingredient into our on the back of the pack. Whereas if you use ingredients which are minimally processed, such as just grinding, right, a flour, we are adding something which consumers are more familiar with. Plus, it has the advantage of bringing unique nutritional elements in, and it has the advantage of being able to bring unique textural and anisotropic elements in.
Okay, we have one more question that says, have you worked on cashew proteins and what were the findings?
Yeah, so I mean, we had a very limited work with cashew proteins, right? Uh, Big cashew uh, processor came to us with their proteins. Uh, the initial idea was, do they texturize, right? They do not texturize well on their own. Uh, but as a blend component, we could do magic with it as well, right? Like most of the proteins. So it has value, but there's so many other uses that you can think of, right? Not just, I mean, texturization is just one application of a protein. In our lab, as I said, we have texturization as a big block, right? We also have extrusion to make expanded snacks and non-expanded applications. We make uh, legume analogs. For example, dal analogs is a very big program, yeah, for midday meals, etc. In our group. Then we have plant-based milks. We also have baking applications, right, and ice cream applications and other applications with, right, whole range of baking that we have now developed expertise in, with high protein bakes, right from bread to muffin to pizza bases to rotis to cakes to brownies, etc., that we can make with them. So the number of interesting applications.
Very interesting. There's one more question that says, can any value addition be done to the soy residue left after protein extraction by using extrusion process?
Oh, we haven't looked at that. I'm sure there are opportunities, but I haven't looked at what is left behind and what's the nutritional profile of it or the physicochemical profile. If somebody has, would love to get hold of it. Please send it over. We'll play with it.
Okay. We'll do a couple more questions before we close. Is there any difficulty in mushroom protein as a major source for texturization?
So we haven't played. PhD student Sahana, I think she's on the call, and so we haven't really played with it in the extruder yet, right? We're looking at it for flavor block, umami flavor block. We're looking at it from a vitamin block, a nutrient block, and we are looking at it from at what levels can we incorporate meaningfully along with other good texturizers, right? We don't intend to make 100% soy uh, textures, right, as such. And right now, at least, we are not into mycelial fermentation, right? That's an altogether different. You don't need texturization. You are creating your mycelia, which is, and then you just cut, chop, and form it into a. So we've not done that, right, yet. But anybody interested in working on that problem, welcome to do a master's or a PhD in our lab.
Okay. And the last question for today, which functional properties, in brackets, solubility, water absorption capacity, or gelling of a protein isolate influence texturization the most?
Uh, I think you certainly require gelling, right? Without gelling, there's going to be no aggregation. So you do require good gelling properties. Yeah. The soy gels the fastest among all the proteins that we have seen, right? And therefore probably texturizes way better as well. Uh, and then I said that we are looking at not just uh, a single protein property. So you require the melting characters, you require solubility because you're adding water and you want it to be soluble at least at a uh, temperature, and depends on what temperature you can solubilize it. Soy for a high moisture, 110, 120 solubilizes very well. P, you require about 130 to 150 for you to start solubilizing well enough. So that becomes a second interesting parameter to look at. The oil absorption from a juiciness perspective, we don't have very concrete data yet, but we feel that's a key. So having some oil absorption is good. But as I mentioned, from a that we have always got better textures when we have two different components we create a blend that we put into our extruder, right? And therefore the question is, what are the two components that you put together, right? Out of the so many infinite combinations you have. And what we find is that the greater the difference in, say, water absorption capacity between two ingredients, right? The we create many more textures there, right? Compared to where the properties are very similar, right? So we are looking always for materials which have different characteristics when we put them together, right? Nutritionally, they are complementary, right? And physicochemically, they are distinct, right? It's still work in progress. It will stay work in progress over the next 5 years as we understand and play with materials and design different experiments. But it's very clear. I mean, if you look at, start looking at, the bands which form when you cool the material, you will get dark light, dark light, dark light bands when you start visualizing them. And you do understand that, hey, they are because materials are coming out of phase and solidifying at different at temperatures, right? So their phase transitions are happening differently, which is what is leading to the different bands that you see and the different striations that you see visually, right? And that, therefore, you do require materials with different characteristics, right? Physical-chemical characteristics for that striation to happen.
Okay. And that concludes our questions. I'd like to let all the attendees know that in case of any queries on research programs or academic programs at TDU, you can write to Dr. Gurmeet. And if you have any other questions about smart protein, please feel free to write to us. The email ID is in the chat box. I'd once again like to thank you, Dr. Gurmeet, for this wonderful session. It was very insightful. I'd like to thank all our attendees for your presence today, and my colleagues Mansi and Harini for their support with this webinar. Thank you.